finally, I will risk myself into hinting at a possible revision of the Shannon paradigm, which by now I'm actually pretty confident it might be one of the good directions after all the discussions that we had. So let's introduce what is molecular communication. So molecular communication theory is a field whereby communication theorists thought about communication instead of using electromagnetic waves and cables and electronics, but by using chemicals. So chemical reactions and molecule transport. And of course, if you follow the very plain communication theory definition of a system, you will have an information source, being that discrete and can be also digitized, of course, or continuous. Then I have an information encoding apparatus that is actually encoding the meaning of the message inside, in this case, molecular properties. And here I have some DNA and some other molecules. Then there is an emission process that somehow initiates, is like the antenna of the system, and initiates what is the transport of information through the space, across the space. Here we, for example, have a molecular motor that takes a molecule or a bacterium that is carrying around a certain plasmid with some information, or simply just molecule diffusion or even pressure difference. Then we have molecule transport. This can be realized in many ways. Think about chemotaxis, think about molecular motors over microtubules, but also simply the diffusion of molecules in the space. Then we have reception and decoding, where usually we have chemical processes that convert the received information into other signals that can be theoretically understood by a destination. Okay, so this is a very general scheme, and this is one of the pillars of molecular communication as a concept. So the very first concept in molecular communication is a substance. And we regard as a substance as somehow the unit, the semantic unit of information. So a substance is, of course, a form of matter with specific homogeneous chemical composition and properties. So if we take out a single molecule out of a substance, we can say that the molecule is the smallest unit of a substance that maintains the substance identity. So it maintains the substance properties, physical properties, and the ability to take part in chemical reactions. So semantically, this might be can be considered the unit of information in a molecular communication system. So if we merge the fact that this molecule has nanoscale dimensions and the chemical bonds have strong forces, so I cannot manipulate molecules by hand, the only way, or the most general way, to manipulate information in molecule is through chemical reactions. And so what we say is that the chemical reaction rearranges the molecule composition and structure, and it's underlying the primary process in a molecular communication system as intended by a communication theorist, of course. It can be also contained in nanoscale dimension. We say that the granularity of information is actually structurally nanoscale. And so molecular communication is also said to be a nano-communication paradigm. Now, the functional element of molecular communication theory, just to summarize, are molecules that maintain, that carry information through their properties, chemical reaction to manipulate them, and molecule transport processes, from the very first basic process, which might be considered molecule diffusion, to higher level processes to carry information, like it might be, for example, chemotaxis, or a human carrying a virus around. So all this again, through the optics of a communication theory. So source, destination, and a link in between. Of course, multiple links will form a network topology, and so on and so forth. So the most basic molecular communication system, even in a very naive way, is a system where I emit molecules from a source, and then these molecules will diffuse away in the space until somehow reaching the receiver. Now, if each molecule carries some information, and the molecules that gets received, indeed, they will deliver the information to the receiver. And in this way, we are actually comfortable saying that this is a telecommunication system because it's communication at a distance, although it's a transport of matter instead of only transport of energy for electromagnetic systems. So, so indeed, we identify diffusion as the most fundamental and basic transport process. But there might be molecular communication systems that are way, way more complex. And in fact, and of course, we studied this process from a communication theoretical point of view. Oh, one thing that I have to say is that, interestingly enough, diffusion, free diffusion in the space, and that was one of the aspect that encouraged research in using diffusion, is that it's a linear channel. And we know that linearity for communication theory and system theory is just a great tool, meaning that I, for example, can have a frequency analysis of the process. And so you can take the spectrum and see how different frequencies are propagated in the space. So that was but it's simple. It's a naive approach. It's just free diffusion. What happens if I have a more complex system like a cardiovascular system? And in this case, we were studying some years ago the cardiovascular system in terms of propagation of drug molecules, and so it had also an application, direct application of this study. And what we can say is that this is such a complex system with of course nonlinearity, a lot of other effects like absorption, reaction, advection, and diffusion. Still, of course, you cannot study that analytically. You cannot study the spectrum of this channel, but you can still simulate that and so have an idea of somehow how it behaves. So, okay, molecular communication seems interesting from the theoretical standpoint, but how do we, and so yesterday it was mentioned that, for example, through the years, researchers have built even platforms to test molecular communication devoid of all the biology, using, for example, the spread of volatile molecules like alcohol, and then an electric nose or sensor to actually sense them at a distance. Okay, and all seems nice and elegant, but when we want to use molecular communication for a specific application, the direct consequence is that we are thinking about systems where molecular communication is important. And so biology is naturally the system. And so through this overall scheme, we see that molecular communication, indeed, as a paradigm, can be applied to study systems at different scales, both natural. For example, this is just an example with the human body, but you could have an example with any system. So at the level of a system, organ, and tissue scale, like we did with the cardiovascular system, at the level of cell, at the level of organelles, or anyway single cells, as we will see in a couple of slides, and then at the molecular scale. And then from the synthetically engineered system standpoint, we have synthetic biology, where we can actually program molecular communication. So not only we can change the channel, we can also design transmitters, receivers, using synthetic biology or reuse natural ones. And of course, there's the idea that we can create molecular communication devices, and in some cases, we can also interface them with legacy systems. Finally, until going to a grand vision, which is having a network of interconnected devices that are both biological and electrical, which can be actually controlled. It can be forward engineered and controlled. Okay, and so think about the extension of the Internet of Things that we have now in our home, like interconnected thermostat, interconnected laundry, and so on, but having instead part of our body that have implanted devices that are interconnected. And so this is for now a little bit going to science fiction, so I don't want to say that everything is done, but it's a vision. We are preparing for this vision, for this future vision, our theory, okay? Thinking about having some theory before actually doing experiments, and that was something that we discussed this morning. This is one aspect that I've been working on, which is understanding how we can estimate information that goes through a cell. So why do we say through a single cell? Well, we said that it's inherently difficult to know all the processes that are inside the cell, but also we thought there was something that we came up in these 2 days, which is the time and evolution scale, and the difficulty in embracing that with the current theory that we have. And so our very first approach to this was like, okay, can I consider a cell, but not within an experiment, but just the cell as a concept, single cell, without the, of course, reproduction and evolution of it, but just consider what we would call an asymptotic cell. Okay, so I have a cell, and asymptotically, it's an organism that behaves in a certain way, but without the evolution per se of the system. And so, if I have a cell and I have this kind of black box, in principle, I can define signals that are outside of the cell as code words. And so I might have signaling molecules, metabolites, environmental stress sensors. And this can be considered the input information. So what is the output information of an asymptotic cell? So we have several processes, okay, but let's imagine that we don't know all about that. Actually, we don't know much about this process, but at least I can tell something about cell growth. I can tell something about proteins that, for example, are released by a cell, or even apoptosis processes. So that if I change one or more of the inputs, these outputs change. So previously it was mentioned in one of the discussion, one famous sentence from Gregory Bateson, who is one of the fathers of cybernetics, which is information is a difference that makes a difference. And so here we are looking at an asymptotic cell, and we are saying everything, every factor here that is subject to change is actually carrying information inside the cell. And then this information translates in a certain cell behavior, like growth, like the, for example, secretion of certain chemical compounds, or even the cell death. And so what we started to see, and this is just a preliminary work and something that I really did quite recently. We opened up a cell, we divided a cell into different compartments, so a signal transduction pathway, gene regulation pathway, and metabolic pathway, and we looked at how the information flows in each one of these compartments. So we started to quantify information from the signal transduction to the gene regulation, quantify information from the enzyme expression to the cell metabolism, and then finally quantifying the information in the integrated cell system. And so to do that, of course, we had the concept of asymptotic cell. We couldn't do experiments, but we took into account that we have the possibility of simulating. And so there is actually one type of simulation that is called flux balance analysis, where indeed you take the genome of the cell and then you put the genome into context. So you tell the simulation engine which chemical compounds will be around the cell, and then you let the cell adapt and make the best out of the system it is now in, depending on its own genome. And so what we did is indeed use flux balance analysis to study this relationship. So how much information can I percolate in a cell from gene regulation to metabolism? And then we actually used another simulation engine to study signal transduction, and we use actually a quite old model, ODE-based model, where we could actually measure how protein-to-protein interaction networks work. And so what we did is we took into account a system where I have extracellular signals. Then I have the model of a receptor. Then this signal percolates inside a protein-to-protein interaction network until showing up into state changes of transcription factor, and then giving rise to a specific cell behavior. And so what we did, we could simulate the system, and if you see here, each protein-to-protein interaction was actually quantified in terms of information flow through that particular interaction. And the measure that we use is simply mutual information. So how much information goes through a certain interaction between molecules in this system. And here, just to exemplify, we divided the system into 3 different blocks. One is the information source, so certain extracellular signals. Then I have my channel, which is the protein-to-protein interaction network. And finally, I have the information that percolates into the transcription factor. And we represented here mutual information, of different values of mutual information with different sizes of these nodes. So we took this model from this paper, is a quite old but established model, JAK-STAT pathway. And what we did is we just placed ourselves at the input and output of the system, and we computed an empirical mutual information. So we just took all the values of input and all the combination inputs outputs, and we applied the mutual information Shannon analysis. And so we could, in this case, and it's a very, very preliminary result that doesn't have any pretense to be a general, but it's just a methodology. We understood that from a mutual information, actually from an entropy, so information content of a source that actually the input signal that we were changing, that was the interferon gamma in this case of a 4.35 bit, we ended up having an information content of the activated state of the dimer STAT1N, which is a transcription factor of 1.65 bits. And so this is just, I cannot go through that given the time, but this is a table where we reported all the mutual information values of different nodes of the network that are actually reported here on the graph. And so we did two analyses, one that takes into account the maximum value that we have in output as my output signal, and the other that takes into account all the time evolution containing information. So the second somehow first principle approach that we used was related to metabolism. And so what we did here, as I mentioned before, we actually focused on the flux balance analysis of a simulation engine, and what we wanted to demonstrate is if we ignore for some time all the signaling processes that are undergoing in the signal transaction network, and we just take into account the metabolic processes, and we have a series of chemical compounds which might be metabolites or other type of signals that constitute a code word. Can I characterize a cell starting just from the genome of the cell, so just the genetic code? Can I characterize a certain genetic code of a cell in terms of information bits that can go from input to output, only using a simulation-based approach? So I just take the genome and I have a certain routine that actually includes the flux balance analysis to actually understand how much information goes through a cell. And so we did that, and I don't have here all the details of the equation, but what we did is we analyzed different stages of the simulation and different input and outputs, and finally we got this table for these two organisms, B theta and M smithii. And the reason why we chose these two syntrophic organisms is that one of my collaborators in biochemistry was actually working on them, and so we could compare these to some experimental results. What I told you so far is a rather naive approach of considering a cell as somehow a black box channel that has inputs and outputs, and trying to estimate using a simulation-based approach the amount of information that can go from the inputs to the outputs, and try to use the number to characterize a cell. We believe that such a number, of course, if further developed, could be a way or a new way to characterize a certain species in terms of how many bits can they, for example, absorb from input to output in their behavior. So the second the other big topic I'm going through today is molecular communication between engineered cells. So cells naturally exchange information through molecules, and the idea is applying molecular communication concept to a system. This is just a prototypical system of two cells communicating through small molecules. So I have here a certain biological circuit that produces reactants, and then I have produce small molecule after some reaction here, and then I have other reactions where they bound to receptors, and then they change the state of transcription factors, and finally they are recognized. And so what we did for this is actually we try to apply similar concept that you saw before to a model of the system to understand how much information can we get from input to output again. And then the second thing that we did is we applied coding theory to actually design biological circuits that could apply a certain coding structure to protect information from the noise in the channel. And so what we did here is we took into account this prototypical system. We tried to have a system at the minimum terms, And what we did, we actually reapplied a similar thought that we had before. So we have a source, we have a destination, and then we want to understand how much information is undergoing between the two. So how much information is delivered in bit per message. So here we had an analytical formulation, and I won't go too much into detail, but what I want to say is that we actually took into account this system. So we actually took real numbers out of the LuxLi-LuxR system, and we developed a simulation based on ODE. This is realized in MATLAB Sim Biology. And then we got numbers out of it. So we actually talked about, so we studied the input-output relationship. We saw that there is a linear phase, there is a saturation phase, and then for different input concentration probability densities, we actually studied how much information goes through the system. And you see that it's not a whole lot at all. And so then the next question was, can we apply coding theory to enhance this performance? And indeed, we did. And actually, out of this work, we received a Best Paper Award in InfoComm, which is one of the major conferences in communication from the IEEE Communication Society. And what we designed is actually we imported the concept of parity check encoding into biological circuits. And so we just built some designs. These are just some constructs that we used. And then out of that, we developed a simulation, again, in MATLAB Sim Biology. And this is the receiver part. And then we studied the bit error rate in terms of performance. Okay, so having said this, overall concept, Internet of Bio-Nano Things, I already presented that to you briefly, is the idea of having an interconnected system where I have biological and electrical entities at the same time, and they might be connected also through natural channels. An idea we are working on here is with actually Greg Payne and Bill Bentley is can we build interfaces between the biological world and the electrical world to enable these kind of applications? And here the Rebionics grant, which is funded by the SemiSymbio program that you heard about before, is actually all about building these building blocks that can bridge biological computation, memory, and communication with the counterparts in electronics. And now a last idea. So we said information is a difference which makes a difference, Gregory Bateson sentence. And so one thing I wanted to say is that when we send an email through the internet does not care and does not change with respect to the content of the email itself. So if the email is a love letter or it's an email of insults, the channel doesn't have any consequences. It's just a consequence for whoever reads the email, of course. So information, somehow we can say that is propagated, but is decoupled from the media physics and chemistry, or anyway it passes through and there is no consequence. But what happens in a biological system? So we of course, you know, everything here, we have the central dogma, we have communication through cells. And what I want to argue is, in this case, information is coupled with the physics and chemistry of the structure itself. So, for example, I have a network of cells, and they are communicating somehow, and then that signal that creates apoptosis passes through. So, from the point of view of the communication engineering, that system is a network, but when that signal passes through, not only gets propagated by the system, but it also affects the system per se, because cells will die out of that message. And so there is a tight relationship between semantics and information semantics and the channel that transports this information, which we don't find in many systems that we study in computer engineering. And so from this, the idea of the wet communication theory grant, where I have the same scheme here as you can find in information theory works, but here I have actually two effects on this box, which is somehow the channel. I have that there is time, and we talked about that many times today. So I have a time dimension that was usually missing. And I also have a semantic dimension because the message is actually modifying the channel itself. This project includes also a wet lab component. This is one of the results of this project. In the first year, one senior design team built this tool that allows to simulate a system and compute information theoretical parameters, and you are welcome to try that out. It's on my website. And then as a conclusion, so molecular communication theory is a viable paradigm to characterize information flow in biological systems. Biological systems and processes are often analytically intractable, and that's why we have to make assumptions and use simulation engines or experiments. Coding theory can inform synthetic biology for engineering biological information flow. And also, Shannon might need some revision. And I offer here a hint, but it's, of course, much bigger than me and much bigger than what I presented. These are just some acknowledgement, some grants through the years, and collaborators and students. These are some publication venues for whoever might be interested. And thank you very much for your attention. I'm sorry for a little bit. And I'm hoping to questions.
So that was fantastic. So I just actually have a technical question regarding the estimates of mutual information that you made for the JAK-STAT pathway.
Yes.
So there, it flashed by really quickly, but I mean, as I understand, I don't know if that thing has a steady state. So the question really is there some sort of integral over time of this thing?
Oh,
so
yeah.
I was just curious, what is the the information between?
Perfect. So that's true. That is dynamic, and so we had to make an assumption, and so the assumption to apply our nice information theoretical relationship was that we present a signal. We present a certain value of the interferon gamma here at the input, and then either we consider the output the maximum of what comes out during the dynamics, and this is all the dynamics that would be observed, or we actually integrate the whole time evolution. Okay? We actually found out that if we integrate the time evolution, we don't get we would be prone to think that we get more noise out of it, and indeed we do, but we also get more information. And the two compensate in a way that when I have time evolution, I wrote these orange values, and they are generally higher. And I should have gotten through that, but it's just very, very, yeah.
Another question? Yes.
Thank you. Max, I'm not sure, or I didn't understand, where you see the difference between the biological system and a multi-user system. In a multi-user system, say CDMA, you have to also pick out the information that is taught for you. And also in biology, you have certain sensing structures that fit to a certain molecule that is on the way. Isn't this the same thing? So there's like a lock-key counterpart,
in
right?
this approach? In this one?
I mean, you're saying it's different in the sense that the semantics matter.
Oh, yes. I didn't mention that. Okay. So this work was done, was kind of preliminary. So this is a very naive approach. But what we are actually working on now is exactly, we are expanding this concept about semantics. And what I mean, oops, I'm sorry. So you mean this concept here?
Yes.
Yes. So this is what we are working on now, and it comes out of our thought after the initial preliminary work that is, as I mentioned, is very naive approach to biology and to measuring information there. But what we are doing right now is actually rethinking that, taking into account the evolution of the system, and really focusing on that limitation that we saw also in many breakout sessions that was mentioned of Shannon with respect to the time and with respect to the semantics. And where do I see the semantics? That's the question. I don't know if I.
Yeah, but I mean, semantics is like, you would say, labeling the information to make sense out of it, right?
Yeah.
You can label also in technical systems, and you do in a way. You can also prioritize information,
yes,
which is also labeling in some way, right?
Yeah, but is that really changing the channel?
It's not changing the channel, but the reception filters out, right? It doesn't change the channel.
Yeah, but in this case, I argue that the channel is also changed.
Okay, this I didn't
at the
get.
beginning.
Okay.
Does that
maybe that I overheard.
does that make sense?
Maybe. I don't know where the biological function is, but this takes longer probably.
Okay, okay, yes, yes.
Just 2 more questions?
Yes. Hey, Andre. Yes.
Hey, great talk. So maybe just to continue a little bit on that, I think message that an example you gave about the cells dying, right? It's going to take a little bit of time. So when you say that there is a message passing through, initially the cells are somewhat open to what the meaning is going to be or what the semantic is going to be. So in a sense, it's sort of within the same context of time being important. So it depends on whether you would like to know what happens now, what happens a little bit in the future, or a lot in the future, right? And
yes.
But another sort of thing you can add to that is that, of course, if you engineer systems, right, you don't need to be affected by that, right? So it really also depends on the application. If you want to understand biology will be changed by the messages that the cells receive, and therefore could not only sort of change the response to that, but change how the future messages will be interpreted. Whereas in engineered systems, you don't need to be affected by that at all, right?
Yes, and I agree with you, and I will answer saying that it might be the case that we want actually to exploit this property as a design feature. So we might want to design a system that actually changes with respect to the message. So, perfect, it's a good hint. Yes, Chris?
Yeah, I may have missed the boat, so that's a preface to the question. Usually, when we're talking about a Shannon sort of system, we're talking about multiple channel uses. The source and channel are independent of each other, and that's why mutual information works. What's different here? I mean, why, you're saying you want to tweak Shannon, but how?
This is the work in progress, and this is how I think we should tweak Shannon. But the approach that I presented before, I agree with you that is a very naive approach where I consider the two processes independent, and I kind of approach, it's a brute force approach on biological models of channels, just coming from communication theory. And I argue that this approach could actually answer finally your doubt and consider a sort of overall correlation of the whole system with respect to the semantics of what I send and then the message. But I argue that Shannon becomes to be a little bit tight.
Yeah, I'm going to push a little bit on that. There are channels with memory, right?
Yes, that's correct.
There are ways of trying to deal with channels with memory. And again, so how do you define your channel use here? I guess that's maybe the fundamental thing. What is one channel use in this paradigm?
Okay, perfect. So one channel use has to come from a functional definition of the source. So what does the source need to get in order to have a certain function that we can define? And so when we define that function, we define also the channel use. if that makes sense. Okay.
Yeah, sure.
I should time myself.
I was just thinking about your change of the channel again, because based on the data. In the technical counterpart, maybe think of moving into the channel an unequal error protection mechanism, say a code or an unequal error protection modulation scheme. Then you could also, dependent on the data, map it to different channels, right? And then you have also a variable channel in a way. Can't you model it like this?
Looking at.
I mean, depending on the importance of data, you also put it into different like bins of the channel that is unequal error protecting code. Say you have video information where the header is very important, and you put it into the high priority channel that is better protected,
Yes.
and the others less. These kind of things, you could see a part of the coding scheme as part of the channel, and then you can model the same things. Probably. It's just a naive idea, maybe.
No, it's great. I just see that differently because when we have a code, it's true that related to semantics of information, we might have a different channel use. For example, a code word that is longer, right? And a code that is, so that makes sense. But I would argue that in my opinion, with respect to the system, is more loosely related to semantics. It's more related to, so for example, after I send that code word, and I send a new code word, the channel go back to its normal state, I would say. Well, unless there is memory, well, this is a longer problem to...
I don't know. I mean, maybe the thinking is too simplistic in the end, that you need more channel variability than you can model with such unequal error protection mechanisms or something like that. Probably. But it's not totally different in the end.
No, it has, okay, yeah, I take your point. I think it's important, and I have to say this is something that is still in the works. So and so.
I mean, it's still interesting. I mean, the generalization of that is pretty interesting. If you can use the data to change the channel, this would actually be the generalization of all the unequal error protection works that we have been doing.
Yeah. So and still, still that can be a viable solution to come up with something.
It could actually have an impact on technical solutions in the opposite direction.
Oh yes, also yes, might be.
Okay.
Thank you.
Thanks.
Thank you so much. Okay, I think it's done. Thank you. Thank you, everybody. Thank you.